Hello, welcome to our webinar. Artificial insemination is steadily increasing in popularity within the equine industry, and consequently, new techniques and technology are being developed to maximise conception rates. Typically, sperm are collected from a stallion, like Spotlight, and promptly diluted in Extender, a nutrient-rich solution that provides sustenance to the spermatozoa, before being placed in cold storage until the time of insemination. Despite being a commonly used practice, the quality and fertility of the stallion sperm after processing and storage dramatically drops in comparison to fresh semen. Whilst the cause of this decline is not fully understood, it is believed that the concentration and source of seminal plasma and a reduction in metabolizable substrates, along with an increase in metabolic waste, may contribute. In 2013, Alison Kaiser and fellow researchers submitted a paper to the Journal of Equine Veterinary Science in an attempt to rectify this widespread problem. A previous journal article by Love and colleagues studied the effect of daily semen cushion centrifugation used to separate sperm from the old extender and subsequent resuspension in fresh extender on the quality and longevity of equine sperm following cooled storage. Dr. Kaiser and her team recognised that the effect of fertility had not been investigated in Love's research, and thus set out to fill this void with their own experimentation. In their study, it was hypothesized that the limitations in longevity of sperm quality and fertility after storage could be rectified by applying Love's technique, and as a result, yield fertility rates similar to that achieved with fresh semen. Before we continue on with our analysis of Dr. Kaiser's paper, it is important to reiterate some basic male reproductive physiology. In a healthy, functioning stallion, spermatozoa mature and acquire potential motility within the epididymis of the reproductive tract. Whilst it is not unusual for up to 30% of the ejaculate to display morphological abnormalities, non-progressive motility, or no motility, increased proportions of atypical spermatozoa will result in a decrease in virility. This may be due to a pathological or environmental conditions in situ, but in this particular case, a decrease in sperm quality and fertility is due to human intervention and prolonged exposure to relatively unnatural environments.
So, a 9-year-old quarter-horse stallion, known for his breeding success, was selected for semen collection using an artificial vagina. The gel-free portion of the semen was collected and diluted in Extender to provide treatment for 3 groups. A total of 34 mares were inseminated with sperm containing 100 million membrane-intact sperm, which was accumulated from a total of 18 retrieved ejaculates. Different interventions were applied to each group, with the focus being on testing different storage techniques and their effects on overall sperm fertility characteristics, including motility and the number of intact sperm following intervention, and sperm membrane intact integrity, such as SMI. Group 1 was the control group, which consisted of 11 mares, which received fresh semen, which was diluted in an extender to 30 million sperm per milliliters, used for breeding within 1 hour from collection. Group 2 consisted of 11 mares and received cooled semen containing 50% seminal plasma, which was stored for 4 days, semen diluted at a 1-to-1 ratio in extender and subjected to cushion centrifugation, with resuspension to 30 million sperm per milliliters in extender containing 50% autogenous seminal plasma and stored in 15 milliliter plastic tubes for 4 days at 8 degrees Celsius. And Group 3, which consisted of 12 mares, also received cooled semen containing 50% seminal plasma, but was stored for 1 day until it was then centrifuged, resuspended in fresh extender containing 10% seminal plasma on days 1 to 3. The semen was diluted at a 1-to-1 ratio in extender and subjected to cushion centrifugation, followed by dilution to 30 million sperm per milliliters in extender containing 50% autogenous seminal plasma and stored in a 15 milliliter tube for 24 hours at 8 degrees. At regular time intervals, it was subject to cushion centrifugation, resuspension, and further cooled storage in fresh extender containing autogenous seminal plasma. Computerized semen analysis was used prior to insemination, as well as immediately after collection, an extension for fresh semen for groups 2 and 3 to assess sperm motion characteristics and sperm membrane integrity. The percent of total sperm motility, progressive sperm motility, curvilinear velocity, and membrane integrity of the sperm, as well as the pregnancy status of the mares, were measured to evaluate the group differences.
Prior to insemination of the trial mares, the 3 groups of semen were tested for motility and morphology characteristics, of the most important being the SMI count. Group 1, with fresh semen, demonstrated the most favourable characteristics of all 3 groups, with 81% SMI and 76.5% motility. In accordance with the hypothesis, Group 3 provided slightly superior characteristics over Group 2. Given these differences in findings, it would be assumed that mare pregnancy rates would differ. However, this was not the case. Following insemination, it was found that all mares from each group had a remarkably similar conception rate. This led the authors to question the relative importance of the semen attributes in the success of conception, concluding that the motile fraction may not be as crucial as first theorised. The results, while they answered the research question, ended up generating more inquiries than they resolved, including the importance of the phenomenon of capacitation in the acquired motility of the sperm within the female tract. This is a process in which membrane-bound proteins are removed from the surface of viable spermatozoa once they have entered the female tract. However, not much is known about this process and therefore didn't really leave the authors with much clarity. The paper presented several flaws which had much potential to skew or make the results somewhat unviable. Firstly, while other reference papers by Wilhelm and colleagues had used multiple stallions in their studies, this study used only one. Although this was done with the intentions of minimising scientific error by testing different animals, it caused the inconclusiveness of the results retrieved, due to them only being specific to one sire. This results in the assumption that all stallions would produce equal sperm numbers with equal longevity under different conditions, which obviously is not the case in real-world scenarios. Additionally, the sample size of mares in the study was too small to generate enough conception variability between groups, resulting in all conception rates appearing the same. With a sample size this small, it would require vast differences in conception in order for it to appear as a worthwhile result statistically.
As a direct consequence of the aforementioned results, the study concluded that evaluation of sperm membrane integrity is an important tool for assessing the quality of cool-stored semen. Cool-stored semen that was centrifuged and resuspended in fresh semen extender daily was observed to have improved motion characteristics and membrane integrity after 4 days. However, despite improving the sperm characteristics, the pregnancy rate was shown to be similar across the 3 study groups, indicating there is no direct relationship between fertility and sperm characteristics. Clinically, the study may be considered important for the storage and handling technique of cooled semen for reproductive specialists studying the characteristics of sperm. Even so, the importance of this study can be judged as somewhat limited in regard to the improvement of reproductive success. The study itself does not replace or add to current knowledge in the field of artificial insemination, but does work to consolidate the previous findings of semen motility characteristics by itself as a poor indicator of fertility.